Thank you, Professor Naveau, and thank you, Tali, for the invitation to come to this wonderful meeting. I first met Tali about 4 years ago at a meeting in the US, and it's been wonderful to see her quiet, confident leadership build to an organization that's put this meeting together. So it's congratulations to you and your team. So my name is Michael Binks. I head up early clinical research for rare diseases in our R&D group based in Cambridge, Massachusetts. Pfizer, you'll be aware, is a big company, a big American company, famous for Viagra and Lipitor, and you might wonder what on earth we're doing in talking about rare diseases. But in fact, we have 22 products licensed for rare diseases around the world. And a few years ago, Pfizer committed to rare disease as one of 6 therapy areas for focus in the company. And you can imagine that that brings a lot of resource and focus both in discovery and development and around commercialization to the rare disease space. And Duchenne is one of the areas that we really are very keen to focus on and develop therapies for. So what I wanted to do here today was just walk you through 2 of our ongoing clinical programs in Duchenne muscular dystrophy. So you're aware of their the therapeutic hypotheses and their current status by the end of the talk. So first of all, I'm an employee of Pfizer, and I need to disclaim that. I'm not going to read it out to you. Domagrozumab is a monoclonal antibody that has been developed to neutralize the biological activity of a small protein hormone called myostatin. Now, myostatin's normal function is to regulate the growth of muscles. And we believe it may have an activity in Duchenne muscular dystrophy, and I'll show you some of the data that provides the rationale for clinical development of this antibody. We believe it has the potential to slow down disease progression regardless of patient genotype. And we have an ongoing phase 2 clinical study and an active open-label extension study in 10 countries around the world. The hormone itself binds to a receptor called the activin R2B receptor, which signals through multiple pathways to suppress muscle growth by reducing protein synthesis and blocking protein degradation within the myocyte. It was identified in the late 1990s, and very soon after, investigators interested in muscular dystrophy started examining whether there may be any biological potential for myostatin to contribute to the phenotype of Duchenne-type disease, examining MDX models, first in a knockout situation and then with a therapeutic antibody intervention. And in both cases, it was possible to show evidence of a normalization of tissue structure with reduced fibrosis and fatty replacement in the muscle. And that's really the observation that drives us forward in terms of the potential in Duchenne. In mouse models, in normal models and normal mice and in MDX mice, the antibody, when administered, increases lean body mass and increases muscle weight associated with an increase in muscle strength as measured by grip strength. You can see in the mouse it's quite crude. We get the mice to grab onto a cage and measure the force required to pull them off. So not really like a sophisticated clinical examination, but it's the best that we can do. We examined the physiology or the pharmacology of the molecule in monkey models, and were able to show a dose-related increase in lean body mass and muscle volume in the monkey. And we've subsequently explored the pharmacology of the drug in normal human volunteers to show that it was not only safe and that we understand the pharmacokinetics and the pharmacology of the drug, but also showed an impact on MRI muscle parameters in a small study of 8 adults. So that provided the impetus to initiate a phase 2 study in boys with Duchenne. So this is our first study in patients, and it's our first study in children. So it had to be fairly carefully designed to ensure that we understood safety as we went through the study and were optimally placed to measure any pharmacological effect in efficacy. So the study involves a randomization to placebo. You've heard some discussion about the pros and cons of using placebo, but what it really does is enable us to be much more confident about the evaluation of some of the functional measures that are required by regulatory authorities to demonstrate efficacy and are rather subject to bias. So using a placebo allows us to remove some of those bias effects from the analysis. So boys were randomized into 3 groups. 2 received active and 1 received placebo. And then after a year, those who had received placebo went on to receive the active drug. And those who had been on the active drug carried on either on high dose or reverted to placebo. That allows us to see something more about longer-term effects and also to define the effects of withdrawal. We needed that group to look at immunogenicity of the antibody, but also to understand how long any beneficial effects would last off treatment. So this study was conducted in boys aged 6 to 16. They had to be able to walk and were on stable doses of glucocorticoids. The drug itself is given as an intravenous infusion over 2 hours, once a month. And we examined 3 dose levels, looking at the safety profile after a 4-month period and then escalating the dose. And after each 4 months, there was also an evaluation of functional measures and of MRI characteristics of muscle. The focus from a functional point of view for this study has been on the 4-stair climb. We felt that it was a better reflection of proximal muscle strength, which our pharmacology may impact, and actually had some better statistical characteristics than some of the other measures. And it's performed very well for us to date. Now, the primary analysis we're just coming up to and we'll be conducting over the next few months. The study is ongoing, so we do need to retain a blind over the data until the study is complete next summer. But it will allow us to plan a little bit for the next steps. The study is overseen by an external data monitoring committee, and I would like to highlight that committee includes a young man with Becker's muscular dystrophy, and he's provided a lot of input into the data that's been emerging from this trial. And it's been very helpful for the other members of the committee to have a patient perspective looking at that data. Okay, so the next steps really are conducting the primary analysis and making decisions about the remainder of the study and the next studies to follow. I don't expect that you'll see detailed data until later next year. So the other project I wanted to talk to you about is a gene therapy program. It's been a very exciting time in Duchenne in the gene therapy world. There have been 3 separate projects initiated in the clinic in the last 12 months. And our program is one of those. It's an area of exciting science and exciting potential, but with a fair number of unknowns and some risk around these types of novel therapies. So let me give you a little bit of a background to gene therapy. It's obviously an approach to replace or repair a dysfunctional gene. It's most appropriate, at least in the early phases, to focus on diseases where there's a known single gene defect, such as Duchenne. And it's an approach that may improve the symptoms or perhaps approach to normalization in disease, although that's not something we're expecting or claiming from this first iteration of therapies. Gene therapy can be administered ex vivo or in vivo, and I'll explain what I mean in a moment. And the term vector is used a lot. I will use it a lot. What it means is a vehicle that's based on the outer protein coating of a non-replicating virus. And that has the capacity to introduce a novel gene into target tissues and induce its translation so that the novel protein is produced. There are a number of gene therapy techniques. It may be about adding a copy of the gene or a modified version of the gene. It may be about inactivating a gene or correcting a gene mutation. I was very interested to hear about some projects starting off in Israel using the CRISPR-Cas9 system to look at gene correction. Gene therapies can be delivered to cells removed from the body, which are then reintroduced into the system, or they can be delivered as modified viruses into the system directly so that they make their way to the relevant tissues and induce gene change. There are a number of different viral vectors that have been used over the decades. I think that adeno-associated viruses, AAVs, are emerging not only as very popular choices, but the 2 licensed gene therapies in the EU and the US are based on this technology. I'm going to skip this, but it just reminds me that Pfizer have 2 more advanced gene therapy programs in the hemophilias, a collaboration with Spark Therapeutics in the hemophilia B and with Sangamo Therapeutics in the hemophilia A. So we're building a lot of knowledge and experience about not only conducting these studies, but how to make the viruses, and how to negotiate with regulators the various hurdles that need to be satisfied to get to the point of a product. Our particular project is based on 2 decades of work by Professor Zhao Zhao and Professor Jude Samulski from the University of North Carolina. Zhao Zhao engineered a miniaturized version of dystrophin that could be packaged inside an AAV virus. As you know, dystrophin is the largest gene in the human genome, and the protein is equally massive. And it was necessary to identify the minimal fragment of dystrophin that would be necessary to get expressed in order to confer a benefit in function. And each of the projects that have started all involve a miniaturized version of the gene. They're all slightly different. It's very difficult to say whether one's going to be better than another, but that's for another day. The mini-dystrophin gene is expressed by a muscle-specific promoter, and the whole sequence has been sequence optimized, codon optimized, we call it, to enhance the expression in muscle, in human muscle tissue. So we've looked at this gene therapy in a rat model of Duchenne. It's an MDX rat model, relatively new model, but it has some benefits over the mouse model that I won't go into today. But we were able to see a very clear dose-dependent increase in dystrophin expression in green here at both 3 and 6 months, both in skeletal muscle and in the heart. Associated with the dystrophin increase was a measurable reduction in the levels of fibrosis. And you can see on the right there that the percentage of fibers that are expressing the mini-dystrophin protein varies between 50 and 90% in most cases. We've also done some work with various constructs in the dog model, and some of those animals were dosed many years ago. I think the longest lived animal was 8 years, and at that time still had evidence of dystrophin expression. But clearly, these very small experiments in dogs are not necessarily representative in any way of what's going to happen in boys. Further in the rat, we were able to see a normalization of creatine kinase following transduction of mini-dystrophin. That's very gratifying to see a biomarker change of that kind. I very much doubt that we'll see something like that in people, because it's much more variable in Duchenne muscular dystrophy than in the rat. We're also able to demonstrate a dose-dependent increase in baseline strength and a reduction in fatigue in the muscle as measured by repeated contraction. So the study we're conducting is in a few centers in the US, and we've now dosed 2 patients, and they're so far doing very well. Certainly no serious adverse effects have been seen in this time. And I just described the study a little bit. We've actually required 3 open muscle biopsies in this protocol. The boys are between the age of 5 and 12, and they have a single treatment and are observed for a 5-year period with a muscle biopsy after 1 year. We're planning 2 cohorts of 6 boys. Each of the boys needs to be negative for an antibody test to prior exposure to the virus. And that's really for safety reasons, but also to ensure that antibodies aren't around to block transduction of the muscle. So I think that's all I can tell you about this project at the moment, but it's going to be a very exciting time for us over the next couple of years to see hopefully the benefits of some of these therapies, but at the very least to see some useful data that will guide future development. So thank you all for listening, and thank you to those people who have taken part in our clinical trials. Thanks.